The tech behind gene editing is really cool, and I'm going to go over 3 of the most common methods of gene editing in this video. The double helix model of DNA was discovered all the way back in 1953. 30-something short years later, and scientists have already found a way to make changes to the genome. The first finely targeted method of gene editing was something called zinc finger nucleases, or ZFNs. Proteins, known as zinc fingers, are manufactured in a lab, and these proteins can recognize and bind to specific DNA triplets. 3 zinc fingers with a bonded endonuclease attach to each strand of the DNA. The endonuclease cleaves the DNA and induces a double-strand break. If a repair template is included, a new gene can be inserted in the break. If there's no template, the gene is deleted. The second method of gene editing is TALEN, which stands for transcription activator-like effector nucleases. So it all started when scientists found transcription activator-like effectors in plant-infecting bacteria. They noticed that these tails were injected into the host cell by the bacteria to manipulate the host cell's gene expression. And scientists found that if they fused tails together in certain orders, you could target specific DNA sequences. Attach a nuclease to the tails and you've got a fully fleshed out gene editing tool. Compared to ZFNs, tails are easier to design and much cheaper. It wasn't too long until TALENs were overshadowed though, as CRISPR-Cas9 came out shortly after. This gene editing method is actually based on a natural protection mechanism that occurs in nearly 50% of all bacteria and 90% of all archaea. These organisms can become infected by viruses that inject their genetic material into them, and this genetic material replicates over and over, which can be fatal for the host cell. This is where the CRISPR-Cas system kicks in. When these prokaryotes become infected with the virus, they splice part of the virus's DNA, or RNA, and store this genetic info in their own DNA as single guide RNA, or sgRNA, creating segments of genetic code called CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. This allows the bacteria cell to remember the virus, and if attacked again, the sgRNA will guide the Cas9 protein to the segment of DNA of the virus that corresponds to the sgRNA, and the attacking virus will be cut apart and killed by the Cas9 protein. Scientists were able to harness this naturally occurring ability by designing synthetic guide RNA to replace the regular sgRNA. The synthetic guide RNA complements the DNA sequence of interest, so it will latch on only to that segment. Then, the Cas9 protein comes on over and makes a double-stranded cut. After the cell registers the DNA damage, it will try to repair it, and this process can be leveraged to make the required changes. CRISPR-Cas9 is the most efficient gene editing tool yet, and it holds a lot of potential for the future. That's all for this video, and if you have any thoughts about this technology, feel free to leave them in the comments below.